Next speaker is Jonathan Larson from the University of Kentucky, the Entomology Department. Jonathan is a doctoral student in the Entomology Department, and he has done some excellent work with honeybees and other beneficial insects. So he will be speaking about environmental conservation through insecticide stewardship. So, thank you, John.
Thanks, Sarah. Thanks for putting the meeting together.
Absolutely.
Is this the clicker?
Yep, there you go.
Cool. All right, good afternoon, everybody. Like Sarah said, my name is Jonathan. I'm a doctoral student over at UK. I've been working with Dr. Dan Potter for the past 4 years, which is a trippy experience in of itself. How many of you know Dr. Potter? You've met him before. I've been doing a lot of work with him and Carl Redmond that has to do with insecticides, the ones that we use in turf grass, and seeing what they do to the non-target organisms that live in turf. So as we all know, a lot of people here in the U.S. want their grass to look a lot like this. They want it to be very beautiful, very green, very lush, kind of a big green carpet that's out in their front yard. Looks like the Yankees could show up at any given moment, start playing catch on the front yard here. But the problem is that there are lots of different types of pests that will come in and attack these beautiful grassy areas. There's white grubs, chinch bugs, crane fly larvae, all these different kinds of things can come in and cause these big brown dead patches like you see here, or small unsightly patches of dead turf, and they can be very problematic for some people. So the result becomes that turf is a very input-driven system. We put a lot of insecticides down, a lot of herbicides down, a lot of fertilizers and water to try and give people the grass that they want and try to preserve the grass and keep it as green as we can. There is a rising interest here in the United States, though, in a more sustainable approach to lawn care application. And it's not just in the home lawn setting or on sports fields, it's also on places like golf courses. There are big initiatives like the Audubon Cooperative Sanctuary Program. Has anybody seen these signs on a golf course before? These are things that are being put onto golf courses to try and give birds more habitat. More golf courses are trying to implement organic, quote-unquote, organic practices on their course to get rid of pests, but to keep the environment safe. And this is just kind of a market trend that we have to deal with nowadays. And the agrochemical industry is responding to this as well. If you go back just a few short years ago, right, 1982, then you look at the chemical class market then, and you only have two choices that you can pick from. This is the high days of adventure with Dursban and Dilox and Turcam and 7. These are the carbamates and the organophosphates. And so this has a whole host of problems associated with it. There's not a lot of choices, so you can get insecticide resistance that pops up very rapidly. These weren't necessarily very healthy for people to be spraying outside in their dresses and pearls like that. They're also not very good for the environment. And since there has been this pressure for the last 20 or 30 years to switch things up, the agrochemical industry has responded. And now we have about 9 chemical classes that we can choose from in turf grass. This includes things like, is this a laser pointer here? Yeah. You can get anthranilic diamides, nicotinoids, spinosins, insect growth regulators like Mach2. You can get pyrethroids and stuff. So this is a much broader range of things to choose from. This cuts down on resistance issues, and it just gives you more choices out in the market. And a lot of these are advertised as being much safer for wildlife and for people to be exposed to. So that's where my research has been trying to come in, is to show ways that turf managers can still use insecticides to give people the kind of grass that they want, but do it in an environmentally friendly way, as well as use environmentally friendly products. And since I'm a bug nerd, as you can tell from the bow tie and everything, I've been working with insecticides, the things that we use to target pests, and I've been evaluating both the lethal and sublethal effects. So we want to know, are these products killing things that they're not supposed to be? And if they're not, are they intoxicating them? Are they getting drunk on these insecticides to a degree that they can't do their job anymore? And we're doing it in the field with realistic exposures, because what's the use in doing it all in a lab? That's not going to tell you what's happening in the real world, right? So I've been focused on 3 products mainly. The neonicotinoid clothianidin, which is known in turf settings as Arena. It's also known as Poncho, I think. Does anybody use Arena, Poncho, clothianidin? Nobody? Okay. You don't have to listen, I guess, for the rest of the talk. Also the combination product, Aloft, which combines clothianidin and the pyrethroid bifenthrin. And finally, Acelepryn, which is the newer product on the market. This is part of the new class, the anthranilic diamides. This was originally a DuPont product. If you purchased it before, it was from DuPont, but they had some issues a couple years ago, and they sold off their specialty products division to Syngenta. So this is now a Syngenta product, and this is the new logo that they came up with for it and everything. This product is very interesting for a few different reasons. One, it gives really good control of pests, but it also presents a new mode of action for us to utilize in turf. Most insecticides attack the brain of the insect. They muddy things up in there, making it poison so they can't use their brain anymore. This actually attacks the muscles of an insect. So in a mussel, there are these things called chlorantraniliprole receptors, and they're essentially gates that open and close and let ions flow back and forth between a gradient. We have them in our mussels, but insect mussels are very specialized. That's why a celiprin will work on them and not on us. And what happens is when they eat this product, it attaches to those chlorantraniliprole receptors, those gates, and it binds them open. So all the ions flow out. They can't move their muscles anymore. They basically get a deadly body-wide Charlie horse, and they perish after ingesting this product. And you can get really good control at low rates of just 0.1 pound of active ingredient per acre. The EPA has started a new program for reduced risk insecticides. This is a new buzzword kind of thing, and this is a new project that they put in. And acelepryn was one of the first products to get this reduced risk status. This just means it's safe for things like bunny rabbits and squirrels and fish and birds to be exposed to. So it's good for wildlife. And it's so safe for people, there isn't even a signal word required on the label. It doesn't say warning or danger or caution on the side, which I thought was interesting. So we've been working with this because when I first got down to Kentucky about 4 or 5 years ago, this had just hit the market. There was nothing known about it whatsoever, what it did in the field, except for kill the bad bugs really well. And that's what insecticides are supposed to do, right? They're supposed to hit those pests, help us get rid of them, but they also might accidentally be impacting things we don't mean to control. And I'm going to talk about several different guilds of insects that live in turf. People tend to think of turf as kind of a desolate place, like it's just the grass and nothing much else. It's a ghost town. But it's really kind of a jungle. There's lots of different weird things that live in the grass, and they make a very interesting and complex ecosystem. And part of them are the natural enemy guild. These are the things that eat the pests that live in turf grass settings. And in grass, it's made up of things like this ground beetle that you see here. These are small brown or black beetles that scurry around in the grass eating things. There's also spiders. Sorry for any arachnophobes out there, but there are lots of spiders that live in turf, including the wandering kind, like this wolf spider. They crawl around very quickly. This one, she's a mama. She's got all her babies on her back right here, and they wander around eating prey items. There are also spiders that are very small. They build very tiny webs down close to the ground, and they also are good at consuming lots of different pests. This is a rove beetle. They're identified by these half wings on their back. It looks like somebody kind of chopped part of them off and then threw them back out in the environment, and they're very good at eating stuff too. But the King Kong, gorilla, Pac-Man of all the predators in the turf grass is definitely ants. These things wander around in big roving bands, eating just about everything in their path. In the south, further south, they have the fire ant, of course, that's really good at doing that, as well as stinging people. But here we have the turfgrass ant, Laceus neoniger. This one you can see is a roaring insect or something, I think, in this picture. And finally, there are the parasitoids. How many of you have seen the movie Alien or any of the sequels or something? Parasitoids are kind of like the aliens in that movie. They sting something, and an egg hatches inside of it, and then it eats the inside of this insect and then pops out as an adult. It's kind of a horrifying thing when you get down to it, but they're really good at controlling certain pest populations. This is a Typhia wasp, which we'll talk more about here in a second, and they attack Japanese beetle grubs. So there are these predators. What good are they to you? It's good that they're there, but what are they giving us? What are they good for? Well, just to throw out some numbers, they're really good at controlling lots of different kinds of pests. Some research that Dr. Potter did, I think before I was born, showed that these predators are really good at reducing things like sod webworm eggs. Things like ground beetles and rove beetles wander around in the turf, and they find the eggs for these ugly little caterpillars, and they can reduce them by up to 75%. And that can help you to keep these pests below their economic injury threshold, right? So they don't get to that population number where they start causing evident damage to turf. Ants, like I said, definitely the big time predator that lives out in turf settings. You can see a pack of them here taking down this caterpillar. They're going to cut it up and turn it into caterpillar hot dogs back at the nest. But they're really good at their job. They can lower grub populations by 20%. They eat the little grubs when they first hatch. They also eat the eggs. They're kind of like the raptors in Jurassic Park. They rove around and attack lots of things. But they're also really good at controlling not just grubs, but caterpillars, like I showed in that other picture. They wander around, they find these guys, and these are just big, fat, juicy morsels of food to them. And they can control up to 62% of these pests out in the field. They eat the eggs, they eat the young caterpillars, and then you don't get as many of these moths flying around. And I mentioned Typhia, but I didn't throw out any numbers about it. These are really good at controlling Japanese beetle populations. We've seen a bit of a decline in Japanese beetles here in Kentucky in the last few years. Has anybody else noticed that we haven't had them flying around as much? We've got a lot of different ideas about what's going on in the lab. We can't prove any of them because we're too busy with other stuff, but they are a sicular organism, so they might be coming back here soon, so don't breathe too easy yet. But these Typhia wasps are part of the reason probably that their populations have been so down. This is a baby Typhia right here. They're basically vampires. This used to be a grub, and it's attached to this grub, and it just sucks the life out of it as it grows, leaving behind a lifeless husk in the soil. And they can control over 50% of these things out in the turf grass. They're very beneficial in that way. A couple of other reasons that you want to keep all these predators around in the turf grass setting is they help to keep pest resurgence from happening, as well as secondary pest outbreaks. Pest resurgence is when if you go in and you spray out an area, you kill everything that's there. If you used to go out and use Dursban or something, you'd kill the pests, but you'd also kill the beneficials. So now there's nothing left here to protect this grass, and the pests are going to find it again. It's just a buffet for them to come and find. And so this is an area that chinch bugs have resettled in and totally annihilated. And secondary pests are things that we don't control with insecticides anyway, and when we get rid of our main pests, the secondary pests suddenly explode. They don't have any more competition left. So we wanted to know what those 3 insecticides were doing to these beneficial predators in the field. I mentioned the 3 insecticides are Arena, the nicotinoid, Aloft, which is the combination product, and acelepryn, which is the anthranilic diamide. So in order to find out what was happening, we went to a few golf courses and got permission from them to treat large patches of grass out on their golf course. And we put these pitfall traps out there. They're just these little cups or tubes that you put into the ground. And there's ethylene glycol in there that kills the insect when they fall in. And it's just a way of measuring how active they are in these turf areas that we sprayed with insecticides. So we catch a lot of predators with that. And we also put out sentinel prey items. We wanted to know if they were still able to eat things. This is called an egg stick. It's just a big popsicle stick, essentially. And down there on the end, you see this white swatch of cloth. There's a bunch of black cutworm eggs that are attached to that. And we leave these out there for 24 hours and let them come and eat those and see what happens. And the results of that experiment are seen here. We put these egg sticks out at 1 week and 3 weeks after treatment, and we found that the combination product really knocked back predation rates. You can see it's lower than 50% at both 1 and 3 weeks. The clothianidin, the neonicotinoid product, it didn't do anything in comparison to the control, but compared to acelepryn, the newer product, it has a lot lower rates of predation. So these predators are sick that are still there. They're not very active. They're not eating the things that they're supposed to eat anymore. But it's a resilient system, right? Things bounce back. Things get better. So at 3 weeks after treatment, we're already seeing things come back, except for in this combination area. They're still pretty sick there. In the end, after looking through my pitfall traps and doing these sentinel prey items, we found that chlorantraniliprole had no impact on predator numbers. There are still bunches of ants and rove beetles and ground beetles in these areas, and they were all doing their job. They were carrying away things that they like to eat, like this little guy here. But the clothianidin and combination product both had negative impacts on these guys. The clothianidin reduced their numbers, and when it was combined with the pyrethroid, it reduced their predation rates. So if you want to conserve these insects to be part of your IPM strategy, which is a really good and efficient way of keeping pest populations in check, you have to consider what you're spraying and when you're spraying it so that you can conserve them. We also wanted to work with Typhia wasps. Like I said, they're really good at controlling grub populations. These are turf cores. These aren't mine. These are from Ohio State. Boo, nobody likes Ohio State. But this is a good picture of what we worked with. So we took these turf cores and we treated them with the 3 products, 1 product on each turf core, and then we cut a hole in the side and we put 5 grubs to live in there, and we let them get situated for a couple of days. And then after that, we caught a bunch of Typhia wasps, like this one here, and then we put 2 Typhia females inside each of these pots that had these cores in them, and we let them get to work, try and find the grubs so they could sting them and lay their eggs in them. And what we found was really interesting. The arena and the aloft, the nicotinoid in combination, they pretty much bombed away any parasitization. They're not very effective anymore. Only 9% were parasitized in the arena cores, 0% across the board for the combination one. So this is simulating if they were, what they do is they fly around on a golf course and they smell a grub and then they just fall out of the sky and then they dig down through the grass and then they sting it. If they're flying around on, say, a golf course that had been treated with one of these, they might fly down and land into a patch of turf that's been treated, and then they get really sick and they can't do their job anymore. We didn't see this negative effect with the Chlorantraniliprole. No impact on survival of Typhia or their parasitization rates. The clothianidin and combination product, negative impact on these rates. The other thing to keep in mind is not just what you're spraying, but when you're spraying it. It's possible that you could continue to use neonicotinoids and combination products. They're very good at what they do. They're very effective at controlling grub populations. There's nothing inherently wrong with them. They're just a tool, right? But if you use the tool differently, you can help to preserve these things like Typhia wasps. This is when they're active in between April and May. As these Japanese beetle grubs, they overwinter as these big honkers like this. They come back up towards the surface to feed a little bit before pupating and turning into an adult between June and July. So the Typhia wasp flies around in May and April trying to find these big grubs like this. And we're often putting out preventive applications from April, May, or June. If you could just adjust the timing a little bit to the end of May or in June or July, you can still have preventive applications to control this part of the pest life cycle, but you're avoiding treating and killing the Typhia wasps. So you can still use the cheaper insecticides if you wanted to do that option. With that, we're going to move out of the natural enemies and move into the decomposers. This is an important guild that a lot of people forget about because they're kind of out of sight, out of mind. Earthworms live underground. Most people think of them as fish bait, or they think they're gross. There are also tiny things like mites and springtails. Springtails are really cool. They jump around. They've got this like spring on their butt that makes them jump really far. And these all are very good at their job, which is decomposing organic matter. Without them, we'd all be up to our ear holes in rotten leaves and rotten grass clippings and stuff. So we have to have these around in order to break things down and add nutrients back into the soil. And we wanted to know if the insecticides we use in turf settings are doing anything to them. And again, we treated big areas of grass, and we took core samples and counted up earthworms, checked how healthy they were, and counted the soil microarthropods. This is one of the earthworms I dug up. He's saying hello. It might be a she. They're kind of hermaphroditic. They can be both. They're weird. The Chlorantraniliprole product did not have any impact on earthworm health. They were all healthy and in high population numbers, or on the soil microarthropods. Mites were still healthy, and so were the springtails. The clothianidin in combination product, on the other hand, reduced earthworm numbers, and the ones that we did recover from those areas, they were sick and stringy. They were like half-cooked spaghetti. They had big lesions on their side. They were very sick looking. And let's flip the microphone. The springtail populations were also very sensitive to these two products. The mites are apparently little tanks. Nothing affects them, so they were fine in all the different settings. But in comparison, between the 3 insecticides, Chlorantraniliprole seemed to be the friendliest for these microarthropods. We also wanted to know if they could still do their job, so we buried these litter bags on a golf course. They were lucky enough to be chosen for us to come out and dig part of their golf course up and bury these out there. These are just, this is some screen that you're supposed to use to keep like squirrels and stuff out of your attic. And we heat sealed it together to make these purse things that you see here. And on this side is some grass clippings, and on this side is a thatch disc. And these were buried for 4 months in areas we treated with our insecticides. And we dug them back up and we saw what happened. And I could show you a bunch of numbers, but a picture is often worth 1,000 words, right? So you can see in areas we treated with the clothianidin, the grass is all still pretty much there. The thatch disc is mostly intact. When the Chlorantraniliprole areas, though, the grass has been decomposed and eaten. The thatch discs are busted. All that's left is soil. So these were still very healthy earthworms and springtails and stuff that could get in here and eat this stuff away before we dug it up. So if you want healthy earthworms and healthy soil microarthropods, Chlorantraniliprole seems to be a friendlier option for them. Now sometimes, I've been talking about beneficials like they're all good all the time, but you have to have balance, right? You can't just have all of one thing in an environment. Just like Darth Vader, sometimes beneficials go bad. He's good in the bad 3 movies and bad in the good 3 movies. So sometimes things like earthworms can take over parts of golf courses, especially. This is a golf course here in Kentucky, and these are all earthworm mounds, or earthworm casts. They come up, this is their fecal material, they come up and cast this out at night. This guy can't even golf. He has to move all of these out of his way before he can even take a shot. And these are being caused by lots of invasive earthworms that are really coming into the United States lately, and they're not being controlled anymore by some of the older chemistries. Ants are another big problem. These get on tees and on putting greens. You can't putt around this. If I were a golfer on this course, I'd get really angry. These can also kill out spots of turf. You can see there we've got like a $50 sized dead spot of turf. And these are starting to pop up more and more as people are switching to acelepryn. We've been getting more and more calls about this because, like I said, you're not accidentally controlling the ants and the earthworms anymore. So they're starting to see outbreaks of these. We wanted to see if that was true, if that was really happening. So we did some trials with earthworm casts. And what we found was that indeed, if you have a cast problem, acelepryn is not going to help you. There's lots of casts still in those areas, even compared to the control areas where we didn't spray anything. There's still a lot of them there. However, aloft and arena decreased earthworm casting by 70 to 81% for aloft, 42 to 56% for arena. Now, this is off-label kind of stuff. I'm not advertising that you spray aloft and arena if you have an earthworm problem. This is just a side effect, a byproduct of their chemistries that they're doing this. And acelepryn isn't some kind of earthworm steroid. They're not bringing the earthworms in by spraying this product, but it isn't accidentally controlling them anymore. Same story with ant mounds. We went to a golf course and treated areas with aloft or acelepryn that had lots of ant mounds. And we found that aloft decreased ant mounds by 90%. Acelepryn had no impact on the ant mounding activity. So lots of these ant mounds were still on these tees here. Now again, it's not bringing the ants in, it's just not accidentally controlling them anymore. I'm not advocating that people switch back from acelepryn to the older products. I think to be efficient, it's best to use acelepryn to control the pests, and then use things like aloft maybe as a spot treatment if you get these outbreaks of earthworms or ants on your golf course. So with that, I'm going to leave the pollinators, or I'm going to leave the predators and the decomposers behind and talk about what I've been doing with the last 3 years of my life, which often gets me in a lot of trouble at meetings like this. I'm going to talk about pollinators. It's a very interesting, charismatic group of organisms. A lot of people care about them. They're made up of things like bumblebees and honeybees. And this group is very important to beekeepers and to agriculture. A lot of our crops are dependent on pollinators. But I've wanted to know what's going on with them in turf settings and if we're accidentally impacting them. And so pollinators are kind of a ship on a stormy sea right now. They're beset on all sides by problems like diseases. There are new diseases coming out of places like Europe all the time that are impacting bees. There's habitat loss. We keep turning natural areas into Walmart parking lots and cornfields. There's less places for the pollinators to build their homes or to forage for food. There are new parasites that can wipe them out. And there's also bad beekeeper practices. Beekeepers often drag their hives from one end of the country to the next, put them in toxic settings, feed them corn syrup instead of honey. So sometimes they're negatively impacting bees. But these things are all combining together to create a big storm of problems for pollinators that's led to a global decline in their population numbers. But a lot of people tend to focus on another factor, and that is the impact of insecticides, in particular the neonicotinoids, which in our world are things like clothianidin, the arena product, imidacloprid. Anybody use imidacloprid? Yeah. Most commonly used insecticide in the world, known as merit usually, or dinotefuran, which is safari. These products are really good at what they do. They control pests really well. They're the most widely used group of insecticides probably in the history of insecticides. They've been widely adopted, and they're really good in comparison to the old stuff like Tercam and Dursban. But to some people, these are a number one suspect in bee death, and they want them to be banned. And we're having to deal with that as an industry, right? Some people are amending their practices. They're trying to change their advertising. You got commercial lawn care companies, Lawn Be Green, and My Green Thumb, Mr. Green Thumb, Be Safe, Organic Lawn Care. This is in Lexington, by the way, 859 number. Yep. And the Golf Course Association is also starting initiatives like this Making Room for Native Pollinators. Things like my old lab mate had a project called Operation Pollinator. Maybe some of you have heard of it before, but these are things that the golf course industry is trying to do to increase pollinator conservation, anything to be a part of the pollinator solution and not part of the quote-unquote pollinator problem. So that's kind of where my research has come in, is providing information on the in-field hazard of the insecticides we use in turf, what they're doing in the long term to bee populations. And I don't just want to point out if there's a problem. I was raised to believe that if you're going to point out a problem, you got to offer a solution, right? It's no good just to say, hey, you're doing something wrong, and then kind of drop the mic and walk away. You got to offer a solution for people to utilize. I really hope that I can pre-arm the industry with some information, put it in a defensible position. These are products, these nicotinoids are going to come under review in the next few years, and I think it's a good idea to have some data to show we're not doing anything wrong, we're not part of the problem, and that we can continue to use these products. We don't want them taken away from us, right? So I've been doing a lot of research with bumblebees for the last few years. We've published most of our results in a journal called PLOS One. This stands for Public Library of Science. Anybody in the whole world can read what we did. This is why we chose this journal, because we wanted anybody to be able to read it. If you go to plos1.org and type in Larson Potter bees or Potter 2013 bees, you'll find our article and you can read it. But I'm going to go ahead and summarize most of it for you today. We use colonies of bumblebees to assess the infield hazard of clothianidin and chlorantraniliprole. After they were exposed inside of these tents, we treated weedy turf that was inside each of these with either of the two products, put the bees in 24 hours after we had treated and irrigated, and then confined them to forage inside of the tent for 2 weeks in the 2011 growing season. This is us putting the bees into the field. This is my former research assistant, Adam, who was like, oh, I'll open the box of bees. That's a great idea. These are basically just shoebox looking things. That's the hive on the inside, this plastic box. It's kind of like this screen here. There's a cardboard box out here on the outside, and on the inside there's a plastic box that the bees live in. This is me releasing them inside of that tent right before I jump out and they start flying around. And while they were out there in the field for that 2-week period, we would go out and check on them and see what was happening with the hives. We counted the number of foraging bumblebee workers inside each tent. This is me learning to count. And we found that in areas that we had treated with clothianidin, there were far fewer foragers inside of these tents. This is a worst case scenario, of course. They're not confined to forage like this out in the field. But if they were exposed to chlorantraniliprole, there wasn't this reduction in workers, and of course the control areas had plenty of live workers in them as well. And we thought this would have a negative impact on their health at the end of the 2 weeks, and what we found was that was right. When we dissected these hives in the lab, we found a higher mortality rate in the arena hives, 34% compared to 6.2 and 6.5% here. So it had a very negative acute impact on them. There's less workers in these hives and less honeypots being built, and honeypots are what they put their food in. So this is a negative result for arena, but again, can't just point out a problem. You have to find some kind of solution. Mowing is, of course, a common practice in turf settings, right? So we wanted to know if mowing would reduce the hazard. So we mowed inside each of our tents, removed those flower heads that we treated in violation of the label, and let new flowers grow in. Once these new flowers appeared, we put new colonies of bees inside the tent and let them forage for 2 weeks as well. What we were trying to test for is if there's a systemic transfer from the soil of the neonicotinoid into the nectar of the clover flowers. It's systemically being transferred into the grass blades all summer, right? That's why these are really good products. We wanted to know if it was happening in clover. And what we found was that it's not. See, there's lots of live bees in these arena hives, in the aceleprant hives and control hives, plenty of honeypots. There were no significant adverse effects after mowing, which we thought was a very exciting result. We believe this shows that just, oh yes.
Any reason why the aceleprant seems to actually
Yeah.
Do better?
I think that's just a fluke.
Just a
of,
fluke?
Yeah.
I just
Yeah, we wondered that too. It's not like a bee steroid or anything. It's not helping them out. But we did have a big hive harvest here, as you can see. Yeah. So again, no significant adverse effects. We thought this was very exciting, and we thought this was good news for the industry. But we wanted to back it up with more results. So for this past summer, I've been doing work with clover nectar, which is about as fun as it sounds. And what we would do is treat these big weedy areas like you see here. And this is full of clover, and we would treat these flower heads and collect the flowers that we directly contaminate, take them back to the lab and get the nectar out. We mowed these areas, let the new ones grow back in, and then collected those. We sprayed these with either imidacloprid or clothianidin. We were only working with neonicotinoids in this project. Take them back to the lab, put them in the refrigerator. You have to do this all in about 40 hours before the flowers go bad. And so I'm in the lab for 40 hours straight a few times this summer, which my boss still owes my wife an apology for, I think. You have to spin the nectar out using a centrifuge, which is just a device that spins really fast. This is Big Bertha, our lab centrifuge. And you spin the flowers really quickly. I picked 300 clover flowers from each pot. I think I collected about 25,000 clover flowers this summer, and spin them around and collect the nectar, and you get a very, very tiny drop of nectar from each clover flower. And then you consolidate it from the individual plots, and these tubes are about this big in real life, and they're only about that full. So this is 300 flowers worth of flower nectar. It's about 110 to 460 milligrams of nectar. So it's a small amount, but it gives us some really cool results. We analyze these with some mass spectrometry. You melt the nectar down, turn it into a powder, and then you turn it back into a liquid and you run it through a computer, and some other guy that's really smart tells me what comes out the other side. And what we found is that there are certain amounts of nanograms per gram of the insecticide in the clover nectar. So all you need to know is the higher the number, the more dangerous it is, the more toxic it might be to a bee. So in the imidacloprid plots, we averaged about 5,500 nanograms per gram of imidacloprid when we directly applied it to the flowers, and 2,400 nanograms per gram for clothianidin. After just one mowing, you can see this is bam, hit the ground, right? 8.4 nanograms per gram, 6.2 for clothianidin. So it really seems to be reducing the hazard. Again, you gotta back this kind of stuff up. This is important. So we wanted to feed it to some test organisms. This is Aureus insidiosus, the minute pirate bug, which is a cool name, I think. We put 10 of these in a petri dish and fed them nectar from directly sprayed plots or post-mowing plots, nectar that was not treated at all. Some of them were given a positive control, which was just application-ready merit, which means it's going to kill them, or a negative control, which in our study was Gatorade, because if it's good enough for Peyton Manning, it's good enough for science. So after 24 hours of eating this, we checked to see how many of them were living, and on average, when they fed on the directly applicated imidacloprid, about 7.5% of them survived. Clothianidin, 25%. Compare that to the control, which is at 83%. That's bad news for those Aureus insidiosus. When they fed on the post-mowing nectar, though, you can see the survival rates have skyrocketed. So we think we've shown that this mowing regime that everybody uses basically in turfgrass anyway is going to help to protect bees from a season-long hazard to neonicotinoid insecticides in turfgrass. The other project I did with bees that you can read about in that article involved looking at their reproductive success after being exposed to these insecticides. What we did was we exposed them to the two products, the Arena or the Acelepryn, for 6 days in those tents like I showed before, but then we moved them to Gainesway Horse Farm, which is a nice horse farm in Lexington. They don't treat their pastures, so there's lots and lots of acres of clover and stuff for my bees to feed on. We put them in this tree line here to develop naturally for a 6-week period. While they were out there, I would go out and check on them. This was the summer of 2012. What does everybody remember about the summer of 2012? Yeah, it was hot. So I'm out there in like 4 cotton shirts and rubber boots and gloves. I got this stupid hat and veil thing on. I'm saying it was hot out there, not very comfortable. I got poison ivy and stung by a bunch of bees. It's a great project. I'm not bitter at all. Left these out there for 6 weeks to develop, and even though it was a lot of hard work and dangerous sometimes, it was really illuminating at the end to bring the hives back to the lab and dissect them at the end of the 6 weeks. This is the inside of a bumblebee colony if you've never seen one. So this shiny stuff right here is the honey, and these are the honey pots that they build to house it in. Dr. Potter and I ate some of this because we're weird scientist people. It doesn't taste half bad, so if you're looking for an alternative. These are where the baby bees are stored. These are housing those as they grow up. And these are queen cells back here where new queens are about to emerge. Bumblebee colonies are a little different. They only pass on their genes if they produce new queens. The new queens are the only part of the colony that survive into the next year. They're not like honeybees where the whole hive survives. So if they don't make these new queens, they're not being successful at all. These are the bees that we pulled out of one hive. They're very angry and stingy in this picture. And these are immature bees. These ones back here are going to turn into queens, these big fat grub looking things here. These are going to turn into workers over here, and these are queen pupa here. This one right here is getting ready to turn into an adult queen. So what we found after the 6 weeks in the field, we weighed the hives as they were out there, kept checking on them. We found when they were exposed to clothianidin, they lost weight at first before bouncing back towards the end of the trial. They never quite caught up with the Chlorantraniliprole or control areas, but they started to gain weight faster towards the end. This is interesting because only the biggest bumblebee colonies will produce new queens. They hit this certain number or certain weight and they start producing new queens. And if they never get there, they're not going to be able to do that. And that is what we found with our trial. The clothianidin exposed hives had only the queens they started out with. Control and Chlorantraniliprole hives, lots of adult queens ready to go out and proliferate in the world. So, to summarize, 6-day exposure to clothianidin treated weedy turf, applied in violation of the label, of course, negatively impacted the foraging activity and therefore the weight gain of colonies of bumblebees and ultimately their ability to produce new queens. Chlorantraniliprole did not have this negative impact, so it seems to be a more bee-friendly option in terms of lawn care practices. Now, we're using these results to go around and talk to people such as yourselves, end users, and tell them that they have to follow label precautions. It's always said on the label for neonicotinoid insecticides, don't apply these to nectar-producing open bloom plants. It's not good for the bees, they've always known that, and if you don't follow the label, you're going to cause issues. We saw that eloquently demonstrated this summer out in Oregon, right? Did everybody hear about the guy who went out and treated the linden tree that was in bloom with dinotefuran? And he killed 50,000 bumblebees in a Target parking lot. And so now Oregon is considering banning neonicotinoid insecticides. So if you don't follow the label, you're going to lose these tools for your use. In turf, the way that you can follow the label is to mow flower heads off either before or after you treat with neonicotinoids to protect pollinators. If you get to a site and it's full of clover flowers or dandelions, tell the house owner they've got to mow, offer to mow for them, charge them for it, make some extra money, or leave them a note that says when they get home they have to mow to get rid of those flowers. You could possibly delay treatment until after the peak bloom of lawn weeds, but you got to get out there, you got to put the insecticides out right. You got to protect these people's yards. That's what they're paying you for. So I think a better option would be to mow the flower heads off. It's pretty simple. It's going to happen anyway. They're going to mow. So that's a simple way to follow the label properly in turf grass settings. The moral of the story is that you can have really great tools, but if you use them incorrectly, you're not helping yourself or anybody else. The neonicotinoids are really good at what they do. They're really effective at controlling pest populations. But if we use them wrong, we're going to impact things that we're not trying to. We're not going to be very efficient anymore. And we're going to lose these products if we continue to do that. So if you use them right, you can keep your insect allies safe, things like predators and pollinators and decomposers. There's new insecticides like Chlorantraniliprole, the acelepryn product that will help us to do this. And there's new approaches. You can change your application timing. You can use granule insecticides. You can use more IPM-based practices to increase your efficiency and help to protect your insect allies. think we all agree environmental stewardship is good. We're all dependent upon the environment for our jobs. We want to give people the kind of grass and landscapes that they want, but we want to keep people and the environment safe. So if we follow some simple guidelines, that's pretty easy to do. All these people were a really big help in completing my research, my lab mates, and people who gave me access to study sites, people who sold me bees. This is our lab motto. I'm going to get a tattoo of it right here before I graduate. And I'd be happy to take any questions you have about insects or insect problems or insecticides.
This video has been part of the University of Kentucky Nursery Crops web series. For more information on the topics discussed, please contact your county extension office.